So, questions? Lab will be closed at 2, not noon today. Is there anybody who needs access to a computer to do your required ID Bachelor?
I have a question.
Yes. Okay. Everybody else okay? I'll probably put one in the room where I had open last time before the exam, just in case anybody needs access to one of those computers. Remember that the other ID backers are not due till the last day of class.
You're going to put that out there too, so if we're in the report to the bot,
Yes, yeah, I'll move it too. Again, it would be helpful if some of you could come early and some of you could come late and kind of stagger the way things we did last time. And so if you want to kind of come on the same schedule you came on last time. We don't have quite as many tie-ups because there aren't as many hands-on things, but it's still nice to be able to stagger things a little bit. So if you would kind of come on your schedule you came on last time, I appreciate that. If you want to pick up some optional points, there will be a guest speaker at 11 sponsored by Scrubs Club. This is the person, the alumni, who is PA, who's an anesthesiologist. And so thanks to Katie for arranging this, and I think there's some food there too, right? Pizza's there too. The universal currency.
Yeah.
Okay, anything else? Okay, we were in the process of talking about how B cells, and as it turns out this is going to apply to T cells as well, become educated in their respective locations. Where do B cells become educated? Bone marrow, T cells, thymus, okay? All B and T cells arise from a type of cell called a stem cell. And then that stem cell will divide and start producing subpopulations of slightly different cells, pre-B and pre-T cells. And we're going to get some further differentiation, and part of that differentiation or education involves learning how to build a receptor and committing yourself to making a particular type of receptor. Each individual cell will make its own unique receptor, and what does that unique receptor pair with? A specific antigen, okay? So for as many different antigens as there are out there in the world, there have to be different B cells, each with a unique receptor. Well, that's a tall order because receptors are made of protein, which are encoded by genes. And on the surface, it would look like you would need a staggering amount of genes to code for just all those receptor proteins to get all of that diversity of different types of lymphocytes you need to respond to all of the diversity of different types of threats out there in the world. But as it turns out, things are not always as they seem. And that is particularly in these cells that are being educated in the bone marrow and the thymus, the genome is pretty unstable. And there are enzymes that can basically select segments that code for portions of those receptors and recombine them and form functional units which are then transcribed into the receptor polypeptide chains. And we call that phenomena gene shuffling. It's analogous to, as I said at the end of the period, having a deck of cards and dealing out 5-card poker hands, let's say. And each one of those cards represents one of the receptor segments. And once you deal a particular hand, then that particular cell will make that particular receptor. Since there are a number of different genes that code for the B and the D and the J and the C elements, then there's a lot of different possible combinations that can occur. So in some respects, the education process that these cells go through, which I kind of liken to being at college, involves card playing. That was a lot of my first couple of years of college education, was card playing. But these cells learn to recognize a particular antigen by building a particular receptor.
This is just like gene splicing.
Yeah. I mean, there's gene snipping and gene splicing. And so that RAG enzyme is going to pop in there and go, well, I'm going to take one of these and combine it with one of these, combine it with one of these, and then I will have a functional message that can be then translated into a functional polypeptide chain. Now, we showed you what some of the possible numbers of different combinations were, just based upon the number of different gene types that code for those receptor elements. And the combinations that we came up with were approximately 5 times 10 to the 6 different combinations. Well, that's where nowhere near the number of combinations that I said we needed and that we achieved to generate 10 to the 23rd different types of B and T cells. So this is explaining where part of the diversity comes from. But where does that additional diversity come from that jumps us from 5 times 10 to the 6th to 10 times 10 to the 23rd? Well, there are some additional sources of diversity. One source of diversity is that RAG enzyme, that recombinant activating gene protein, randomly deletes portions of the D and J elements before it's combining. So once it's snipped those out and it's ready to join together, then it goes and says, oh, I think I'll take a couple nucleotides here and there. Well, that's going to change your amino acid sequence. A different enzyme, couldn't find a name for this one, I'm sure there is one, will do a random addition of nucleotides to the VDJ combination. So once it's stuck together, a V, a D, and a J, then it goes, oh, okay, I'm going to add a couple other nucleotides to that. That's going to change your amino acid sequence. And this is all random. And then finally, those particular cells are very prone to hypermutation. When those B cells are sitting there becoming educated, the genes are randomly mutating while this is happening. So this really jumps up our number of possibilities for combinations. And a result of all of these random and kind of bizarre activities, we generate kind of by chance all different possible combinations. And that's why the immune system can respond to even a completely novel antigen that's been synthesized in the lab that hasn't existed before. Because by random chance, one B lymphocyte with a unique receptor would have been generated by all these random events that would respond to that particular antigen. It is a very creative way of using a limited amount of genetic information to produce all of these different types of receptor molecules, which are central to the immune response. Okay, so what happens once you've started transcribing those polypeptides into heavy chains and light chains? Well, then the proteins start moving through the ER. The proteins actually are glycoproteins, and so there are sugar molecules that are added onto them. So there's some glycosylation that takes place. And eventually, that particular protein is inserted in the cell membrane and is now a dedicated receptor. And as I said, this happens for both B lymphocytes and T lymphocytes. Their receptors are structured a little bit differently, but the same phenomena of gene shuffling produces all the different receptors in those two particular cell types. Now they're educated, they're ready to go to work, so they migrate out into the lymphoid tissue and take up residence. And while they're sitting there, they're in kind of a state of suspended animation. They're not dividing. They're quiescent. Okay? They're dormant. So they've got a pretty cushy job. They put up their feet and they sit and wait to be called to do work. And what's the call to do work going to involve? It's going to involve being challenged by a particular antigen that matches their receptor. And when that happens, that then is going to activate that cell into rapid cell division. So we're going to form whole populations of cells that are identical to one another, a clone. And usually a second signal is required besides activation by antigen binding, usually a cytokine of some kind. And that can then start the cells to start to differentiate into the subpopulation. So for example, a B cell can differentiate into a plasma cell and start secreting antibody. And what's really kind of cool about that is the B cell secretes the antibody with the same antigen specificity as the receptor it had on its surface. It's almost like it starts secreting its receptors now. Some of the B cells differentiate into memory cells. If you're a T cell and you become activated, then you can differentiate into a cytotoxic cell, or a helper cell, or a suppressor cell. In the case of a plasma cell, once that starts producing antibody, there are tremendous changes that occur in the cytoplasm of that cell. And I think I showed you this last time I referred to it. This is a plasma cell that's been stimulated to start producing antibody now. And the combination of the binding of the antigen and some chemical signals from other immune cells causes this B cell to really change internally. And all of a sudden it starts producing a whole bunch of endoplasmic reticulum. It becomes loaded with ER. Because now it's basically turned into an antibody secreting factory. And these cells have a limited lifetime, and they basically just burn themselves out producing and secreting antibody, which is then released into the surrounding environment and will have effects on the pathogens that bear the antigens that cause the antibody to be formed. And we'll talk about what some of those effects are. Now, it is important that we have a mechanism that allows all of these different B cells with different receptors to be deleted if they will react with your own cell's antigens. So one of the processes that occurs after a cell becomes educated is a process that we call clonal deletion. So let's say that we just generate 5 different B lymphocytes here, each with a unique receptor, and that this particular one right here will bind to and react with cell for autoantigens. That's not good. So what does that do? If this recognizes that particular receptor, that sends a signal to the cell, self-destruct. It's going to go through apoptosis. And if this signal isn't properly transmitted, and this cell survives, then that could be a source of cells that could cause autoimmune disease. And that's serious, and that's not a good situation. Each of these other B cells basically is checked out before it's allowed to leave the bone marrow and make sure that it doesn't react with these. See, this one's not binding here. Over here, this one did, so self-antigen. So this cell doesn't recognize self-antigens. It passes the test and now can be released into the circulatory system and will travel to its location. It might be the spleen, it might be the lymph node, or whatever. With T cells, it's a little bit more complicated because there's kind of two levels of selection that take place. One of those involves, will the cell respond to MHC? Because remember, T cells are MHC restricted. And if your receptor will not recognize an MHC, you're no good. You play no role. You're defective. So what's going to happen? That cell's going to go through apoptosis. Cells that can respond then to MHC are further screened to see, do they respond to self-antigen? And if they do, then they are eliminated. And if they don't, then they pass the test. And now they can go out into the secondary lymphoid tissue and take up residence and wait to do their job. So this idea of clonal deletion is really important because remember, we're generating all these different cells randomly. And the likelihood that we're going to generate some cells that will respond to self-antigens is very high. So what happens when you're educated and you're now sitting in a lymph node, let's say, and you become challenged by an antigen? You become exposed to an antigen. Then another phenomenon takes place called clonal selection. So sitting in that lymph node are all of those B cells that have passed the test. They don't respond to self. They respond to foreign. And there's many different kinds of them. They're sitting there dormant. And now they become exposed to their antigen. And likely they're probably going to get another signal from another immune cell that will stimulate the process as well. So what do? Let's say that this is the one that matches the antigen right here. That one has been selected. That cell line has been selected. So now it's going to start dividing and differentiating. And it's going to form a whole massive population of cells that are identical to the original parent cell, all with the same antigen specificity. And if that's a B cell, then it's going to differentiate into plasma and memory cells. And the plasma cells are going to start producing antibody. We'll talk a little bit later on about what memory cells are and how they work. If it's a T cell that becomes stimulated, then it can differentiate into the T helper, or the T killer, or the T suppressor. So basically the antigen is picking which cell is activated, and therefore what cell line or clone starts to divide and proliferate. And again, that's why your lymph nodes are going to start to swell if you have an active infection, because the number of white blood cells is going to increase dramatically as a result of this clonal selection process. Now, most typically, in order for educated cells to become activated, it requires two signals. One signal is definitely required, and that is antigen binding to the receptor. You got to have that happen. Usually there's a chemical signal involved too. A cytokine is involved. Where's that coming from? Some type of other immune cell. I mentioned that T helper cells aid B cells in producing antibody. How? Well, the antigen activates the B cell. That's part of it. But then the T helper cell, which also becomes activated by that antigen, produces a cytokine. And that second signal is necessary most times to start triggering that cell into antibody production. So, what are cytokines? Well, here's some examples of cytokines that we have either heard about or will hear about. Interleukins are a general class of molecules that are cytokines that cause cell signaling among white blood cells, or between white blood cells. So inter means between, leukin refers to white blood cells. When I first started teaching immunology, eons ago, there were probably about 3 or 4 interleukins that were identified, and pretty much all I talked about was interleukin 1 and interleukin 2. There are now 35 different interleukins that have all been identified, and I'm sure that list will grow. You're going to see references to some of these interleukins in some of the later slides. I'm not particularly going to be hung up on you memorizing all the different interleukins and what their numbers are and what they do. But I do want you to know that the complexity of the chemical signaling that takes place between immune cells is really pretty mind-boggling. We've also talked about interferons. Interferons, remember, belong to 2 classes. Class 1 involved stimulating antiviral proteins to be produced. But class 2 were molecules which were immune stimulants. So when we talk about interferons being cytokines, we're talking about the class 2 interferons. There are various proteins that the body produces, which are growth factors, which can stimulate the bone marrow to increase white blood cell and red blood cell populations. So these general growth factors are kind of stimulating cell growth in general, including immune cells. Tumor necrosis factor is a type of cytokine that's produced by various immune cells. It has numerous functions, one of which is to cause necrosis of tumors. One of the things that you'll see about some of these cytokines is they oftentimes have multiple functions. They do multiple things. And then chemokines are substances that attract white blood cells. They are chemotactic factors. And we mentioned chemokines in the context of inflammation. We mentioned chemokines in the context of complement cascade. So I really do want you to be aware of the cytokines and their existence and their importance, and the fact that these are usually what are responsible for providing that second needed signal to complete the activation of either a B or a T lymphocyte. Pretty critical. And this is just a slide that gives you some common cytokines, indicates what their source is, what their typical target is, and what their typical action is. So for example, interleukins were named in the order in which they were discovered. So interleukin 2, for example, we've known about for a long time, and that stimulates a T helper cell, or it's produced by a T helper cell, and it typically causes a T killer cell to become activated and start dividing. Interleukin 4 is also produced by a T helper cell, but causes B cell activation. Here we've got gamma interferon, that's that class 2 interferon that tends to stimulate phagocytosis by macrophages. Here's our tumor necrosis factor, triggers inflammation or apoptosis. And so these are just some representative examples of some of the kinds of cytokines that are produced. Okay. We've been throwing around the word antigen quite a bit. So we need to really dig into a little bit more what are antigens and what makes something antigenic. I said previously that the term antigen is derived from antibody generating or antibody producing molecules. These are the molecules that stimulate B cells to produce antibody. Oftentimes, antigens are considered to be foreign in nature, and those are the ones that we are most concerned about, those that are associated with pathogens like bacteria and virus. But remember, there can be self-antigens as well. And if B cells that react to self-antigens survive clonal deletion and are stimulated, then we have a state of autoimmunity. There are certain requirements for being an antigen, and one of them is size. Typically, something won't be an effective antigen if it doesn't have a molecular weight greater than about 1,000. And that's not a particularly big molecule when you start thinking about it. Another thing that's required is complexity. As it turns out, proteins and nucleic acids tend to be strongly antigenic. Lipids tend to be weakly antigenic. Carbohydrates, so-so. Why the difference? Lipids are pretty chemically simple. They are basically polymers of repeating carbon, hydrogen. There's not much molecular diversity to lipids. I mentioned earlier on when we were talking about endotoxin that endotoxin is lousy antigen, and therefore you really can't immunize effectively against endotoxin poisoning. On the other hand, proteins and nucleic acids are very structurally diverse. Okay? All those sequences of A, T's, G's, and C's gives molecular complexity in DNA. All of those different amino acids in different orders gives you incredible molecular complexity. As it turns out, the receptor on a B or T lymphocyte typically isn't reacting to a whole molecule, a whole polypeptide chain. In reality, it is reacting to a piece of that polypeptide chain, or a portion of it. And that portion of that it reacts to is called an epitope. It's also referred to as an antigenic determinant. So one bacterial protein, like a flagellar protein, can have many antigenic determinants, many different sequences or little clusters of amino acids. And that will then bind to many different receptors. If that happens, we can generate what we call a polyclonal response. The same molecule selects many different types of B cells because a portion of that molecule selects this one, and another portion of that molecule selects another one, and another portion selects another one. So we can have several different types of B lymphocytes activated by the same molecule, a flagellar protein or a cell wall protein. I thought I had a picture of that. Size is also important. There are some molecules that are very complex, but don't have a big enough size to really effectively interact with a receptor and stimulate an immune response. those small antigens are called haptens. They could stimulate, but they're too small. Sometimes what will happen is a hapten will combine with another protein, like a skin protein or a serum protein. And that combination now is big enough and has the necessary size to stimulate an immune response. Anybody in here have an allergy to costume jewelry? Sometimes people will react to costume jewelry and get what's called a delayed hypersensitive reaction. They'll get blisters and things like that from the copper. Well, that's actually an immune response. It's an inappropriate immune response, but it's an immune response. The copper is a hapten. Copper does not have enough size in and of itself to stimulate the immune response. But when it complexes with skin proteins, now that combination is antigenic. There are several different types of antigens that you need to be aware of. And at the end of this list, there will be one and two in particular that will be really important. Autoantigens we've already mentioned. Those are antigens that are self, that still cause an immune response. And these can lead to autoimmunity. There are an increasing number of diseases that we're finding out have an autoimmune basis. But there's still a lot about autoimmunity we really don't know. Why does the body all of a sudden start reacting to self-antigens when it previously hasn't? Because sometimes these autoimmune diseases all of a sudden just pop up. Something like lupus, for example, which is autoimmune, which tends to be developed later in life. We think that there's a good possibility that multiple sclerosis is an autoimmune disease where antibodies start attacking proteins that are part of the myelin sheath and start causing degradation of nerve tissue. So a lot of things there that we don't know. Alloantigens are found in some members of the species, but not in other members of the same species. Remember that I said that all of your cells are covered with these MHC1 membrane proteins that have little bits of your self-antigens displayed on its surface that gives you the cell signature that identifies your cells as being you. So these alloantigens are really important to analyze in the process of tissue typing prior to doing transplantation. Another type of antigen that I have mentioned is the heterophile antigen. And that is the same antigen found in different species. There's an antigen on group A Streptococcus bacteria that is the same antigen you find on heart valve tissue. And if your body starts mounting an immune response against the group A Streptococcus, that antibody can also attack the heart tissue, causing rheumatic fever. We're going to talk at the end of the class about using antibodies to diagnose disease and some of the medical technology that's developed that allows us to diagnose disease through using blood serum and antibodies. And one of the things that you've got to really be careful about in this kind of testing is cross-reaction or cross-reactivity, where you're getting a particular reaction and you think it's due to one thing, but it's actually due to another because of these heterophile antigens. And unfortunately, when you get these cross-reactions, they can give you false positive results. And you know, medically, that getting a false positive can be pretty bad. And if nothing else, it makes you think you've got a disease you don't. But it really can affect treatment options as well. We have also mentioned superantigens. Those are found in both strep and staph. And these are antigens that when they bind to immune cells cause an over-exaggerated response. They cause that cytokine storm where vast amounts of cytokines are released with really devastating effects. Allergens, some of you have allergies, are antigens that cause an immune response, even though the molecule really isn't that threatening. This is an example kind of immunopathology, where the immune response is responding inappropriately. One of the things that we'll see later on when we talk about different types of antibodies is that individuals who suffer from allergies tend to overproduce a type of antibody called IgE, and that then has an effect on the immune reaction to these normally harmless things like pollen. For our purposes, it's going to be very important to distinguish between T-dependent and T-independent antigens. T-independent antigens are ones that can stimulate a B cell by themselves. That B cell only needs one signal, binding of the T-independent antigen. These particular antigens tend to be pretty simple. Oftentimes these antigens are just kind of repeating units of the same molecule. So they don't have a lot of complexity. T-independent antigens will cause a plasma cell to be formed. But you do not get memory cells formed from T-independent antigens. So basically, every time you're exposed to this, it's like you're seeing it for the first time. T-independent antigens are relatively rare. By far, the majority of antigens that stimulate an immune response are T-dependent. The structure of the antigen tends to be more complex. It's not this repeating unit over and over again. If the antigen is T-dependent, in addition to the binding of the antigen, a cytokine is also going to have to be involved to get that cell to do something, to stimulate that cell to start dividing. oftentimes the source of that cytokine is the T helper cell. So if a B lymphocyte gets the appropriate two signals, binding of antigen plus cytokine from a T helper cell, it's now going to start dividing and forming plasma cells and memory cells. So this is where you get your memory response. Why do we get the memory response? Because of the involvement of these cytokines. And we'll talk a little bit later on about what memory cells are and how they differentiate from plasma cells. This is the basis of immunization. So would it be effective to try to immunize somebody with an antigen that was T independent? No, you wouldn't get a memory response. You would lose that particular advantage. The other type of molecule that is incredibly important in the immune response is the antibody. Now, remember, only B cells that have been properly stimulated, plasma cells, can produce antibody. T cells don't produce antibody. The structure of the antibody is very similar to the B cell receptor. The model of antibody structure that we're going to use to start out this discussion is shown up there on the board. And this is a particular class of antibody called IgG. Now we're going to see in a minute that there are more than one class of antibodies. There are actually 5 classes of antibodies. So we're going to start out our discussion of antibody structure in the context of this IgG antibody. So does this look familiar? It should. This looks pretty much like the B cell receptor, doesn't it? Like what was stuck into the membrane. It's got 2 heavy chains and 2 light chains. And those are actually made out of genetic elements. The V elements here are called the variable elements, and the C elements right here are called the constant elements. What does variable refer to? Amino acid sequence. What does constant refer to? Amino acid sequence. For all of the different antibodies within a different class, the sequence of these C regions is the same. It's kind of like a common building structure for all antibodies of that particular class. But for every different type of antibody produced by every different type of lymphocyte, each of which has a different receptor, the variable regions differ. These V regions right here and right here constitute the antigen binding site. So lymphocyte 1 produces a particular type of antibody because it is a particular type of receptor. Lymphocyte 2 produces a different antibody. Lymphocyte 3, a different antibody. Now, if they're all IgG antibodies, this portion in blue is always the same. But this portion here in pink, the binding sites are different. This particular antibody will bind 2 molecules of antigen. Remember the term I used for receptors, talking about how many molecules they can bind? This has a valence of 2. What we're going to see when we look at some of the different classes of antibodies is different classes of antibodies have a different valence. They can bind different numbers of antigen molecules. Why is it important to bind more than one antigen molecule? We're going to see later on. But the immune response would be particularly limited if one antibody can only bind one antigen. So what is all this stuff down here, all this constant region doing? Basically that constant region, and particularly this region right here, that's part of the 2 heavy chains called the FC region, basically defines what that class of antibody can do. Different classes of antibodies have different general functions. For example, you're probably all aware that some antibodies can be secreted. Mothers can secrete antibody and provide that to their infant through breast milk because they produce a particular type of antibody called IgA. Not all antibodies are secretory. Some antibodies can cross the placenta, but others can't. Some can attach to complement and initiate the complement cascade, but some can't. Some are opsonins, the kick-me-signs, but some aren't. What defines what your class of antibody does? The sequence of amino acids in this FC region right here. That basically is different for each different class of antibody. So what we'll pick up with next time is looking at the different classes of antibody and kind of seeing what they do and how they're different from one another in their general functions.